Paxlovid. The U.S. Food and Drug Administration approved the oral antiviral Paxlovid, nirmatrelvir tablets and ritonavir tablets, co-packaged for oral use, for the treatment of mild to moderate COVID-19 in adults who are at high risk for progression to severe COVID-19, including hospitalization or death. Paxlovid is the fourth drug and first oral antiviral pill approved by the FDA to treat COVID-19 in adults. The efficacy of Paxlovid was primarily supported by the final results of the EPIC-HR clinical trial. EPIC-HR was a randomized, double-blind, placebo-controlled clinical trial studying Paxlovid for the treatment of non-hospitalized symptomatic adults with a laboratory-confirmed diagnosis of SARS-CoV-2 infection. Patients were adults 18 years of age and older with a pre-specified risk factor for progression to severe disease or were 60 years and older regardless of pre-specified chronic medical conditions. All patients had not received a COVID-19 vaccine and had not been previously infected with COVID-19. Paxlovid significantly reduced the proportion of people with COVID-19-related hospitalization or death from any cause through 28 days of follow-up by 86% compared to placebo among patients treated within 5 days of symptom onset and who did not receive COVID-19 therapeutic monoclonal antibody treatment. Pharmacodynamics. Nirmatrelvir is administered alongside ritonavir, a potent inhibitor of CYP3A enzymes, in order to inhibit its metabolism and increase plasma nirmatrelvir concentrations. While therapeutically beneficial, the use of ritonavir poses a significant risk of drug interaction due to its potent inhibition profile. Patients and clinicians should consult the prescribing information for Paxlovid, nirmatrelvir and ritonavir to evaluate any potential for drug interaction with existing medications prior to the initiation of Paxlovid. Mechanism of action. Nirmatrelvir is an inhibitor of a cysteine residue in the 3C-like protease, 3CLPRO, of SARS-CoV-2. This cysteine is responsible to the activity of the 3CLPRO of SARS-CoV-2 and potentially other members of the coronavirus family. The 3CLPRO, also known as the main protease or non-structural protein 5, is responsible for cleaving polyproteins 1A and 1AB. These polyproteins contain the 3CLPRO itself, a papain-like, place, cysteine protease, and 14 other non-structural proteins. Without the activity of the 3CLPRO, non-structural proteins, including proteases, cannot be released to perform their functions, inhibiting viral replication. Absorption. The median Tmax of nirmatrelvir, when given with ritonavir, is 3 hours. After a single oral dose of 300 mg nirmatrelvir and 100 mg ritonavir in healthy subjects, the Cmax and AUCINF of nirmatrelvir were 2.21 micrograms per milliliter and 23.01 micrograms hour per milliliter, respectively. Benefit. Paxlovid was observed in patients with prior immunity to the virus that causes COVID-19. Among patients in EPIC-HR who were antibody positive at trial enrollment, the risk of COVID-19-related hospitalization or death from any cause during 28 days of follow-up was 0.2%. Among the 490 patients treated with Paxlovid compared with 1.7% of the 479 patients receiving placebo. EPIC-SR was another clinical trial that enrolled vaccinated patients with at least one risk factor for progression to severe COVID-19. Although not statistically significant, among these vaccinated patients, there was a reduction in the risk of COVID-19-related hospitalization or death from any cause. Because of the importance of reducing the risk of significant drug-drug interactions with Paxlovid, the approved label and authorized fact sheet for healthcare providers for the Paxlovid EUA come with a boxed warning with instructions for prescribers. Prescribers should review all medications taken by the patient to assess for potential drug-drug interactions and determine if other medicines that a patient may be taking require a dose adjustment, interruption and/or additional monitoring. Prescribers should consider the benefit of Paxlovid treatment in reducing hospitalization and death, and whether the risk of potential drug-drug interactions for an individual patient can be appropriately managed.